Well, hello everyone, and welcome back to Science in the Age of COVID-19. Today we are super excited to have another pair of Double Trouble speakers. We have Chloe Villani and Alex Shalek. They will be speaking about the broad topic of single cell atlases, mapping all the cell types that are involved in the infection and progression of SARS-CoV-2. Chloe is a professor at Harvard Med School and at the Broad Institute, which focuses on immunology and inflammatory disorders. And Alex is in the MIT chemistry department, focusing mostly on cancer and immunology. And Alex will go first. Okay, take it away.
Thanks, Lauren, and thanks so much for having us. Let me just make sure that my slides come up and that you all can see them. Everything look okay on your end?
Yep, looks great.
Great. So thank you all so much for having us today. It's my pleasure to co-present with Chloe on some of the work that we've done over the past year plus now, trying to leverage single-cell genomics to understand aspects of COVID-19 disease. And as you know, by the very nature of the fact that we're having this seminar, COVID is a global pandemic, and I don't have to emphasize how many cases, deaths, and vaccines have shown up. There have been multiple different trackers that have helped us to understand the nature of the pandemic and where we stand. Fortunately, we seem to be almost on the other side, but I'm a glass half full kind of person. And hopefully the trend will continue in the improving direction, despite all the concerns that are arising most recently. Sort of starting early in the pandemic when stuff started to arise, there were a number of clinical case reports that came out that started to describe some of the aspects of COVID-19 that I'm sure you're all very well familiar with now. Symptoms in the upper respiratory tract and the gastrointestinal tract, and some general things like fever and malaise. And as as we began to understand what was happening clinically, there were also a number of studies that were done in labs by a number of different scientists that began to understand at a molecular level what was happening in COVID-19. And it was really that, discovered that SARS-CoV-2, the virus, binds to this receptor on the surface of cells, ACE2, and uses an accessory protease, primarily TMPRSS2, to gain entry into the cell, where it goes on to go through a normal viral life cycle to replicate itself and produce new virus. And right at the start, like many of you, we were very inspired to try and do whatever we could to help, but we weren't exactly sure where to begin. And one of the first things that we thought we might be able to help with was trying to identify which cells would be likely targets of SARS-CoV-2, and to what effect, helping us to tie some of these general symptoms that we were seeing over here to really what was happening at a molecular level. And the reason that we thought we might be able to be useful in this capacity is that, we had these huge repositories of published and unpublished single-cell RNA sequencing data. So, these beautiful hairballs like this one that I'm showing you over here that represent multiple different tissues from which we could begin to extract what would be likely targets. And just to help you orient you on this, I'm sure many of you have looked at single-cell data sets before. I just want to go briefly through how these are produced and why we thought they would be useful. So in this particular instance, what we're looking at is a non-human primate where we're looking at 12, 13 different tissues. And so in these single cell workflows, what you traditionally do is you would take these tissues, cellularize them to get the component cells, and then isolate individual cells using one of many different technologies, whether it's a multi-well plate, a nanodroplet, a picowell, or the like. In this particular instance, for this data set, I'm using a picowell device where I'm capturing individual cells with uniquely barcoded beads that append social security numbers to the information from each cell so that we can then reverse transcribe, create one sequencing library, but use these barcodes to figure out what belongs to whom. So basic idea in this workflow is that you isolate the cells, you lyse them, you use oligo-DT primers to capture polyadenylated messenger RNA, you amplify, you sequence, and then you're basically getting information on all of the roughly 30,000 genes that the cells express, but we're getting that for each individual cell. So we're getting billions of data points that we can begin to use to understand really the cellular basis of complex biology. And so once we have all this data in hand, the way in which we make these hairball plots is through some of these complicated computational analyses that basically take out things that are low quality, find things that are variable, and then create structure. And so for the hairball I showed you at the beginning, this is sort of a graphical representation of iterating through this process of taking these data points and pulling things that are similar together and pushing things that are dissimilar apart. And you can see as we keep on iterating through, we converge to a point where we're going to end up with individual points, which are cells, neighboring one another that are similar, and distinct clusters that arise. So to really annotate what I've been showing you so we're all on the same base, this was the hairball at the beginning. I've colored tissue cells by tissue of origin. Each point represents a single cell. And then what I can do is I can go and look at, for each of these individual points, the expression of all the genes within those cells, so the full transcriptome. This helps me to identify what are the different cell types and to figure out what they might be expressing. So with this data in hand, you can probably now understand why we thought we might be able to do something that would be useful in the context of COVID-19, because we could basically go back to these data sets that we already have and use them as a lookup table to figure out which cells might be expressing ACE2 and TMPRSS2. So in those 12 tissues that I showed you, down across the bottom here, what we can find is that there are a number of individual cells which are points in, for example, the ileum and the jejunum, parts of the liver where we see, or parts of the intestine where we see symptoms, showing expression of this entry receptor. Similarly in the lung. But we can go beyond this. Instead of just saying, where do we see expression of these individual, these individual RNAs by tissue, to really figure out within tissues that are impacted, like the lung, what are the different cell types and which ones have not only ACE2, but TMPRSS2. And so here, I'm showing you that as we dive into the lung of humans and non-human primates, we found that really ACE2 showed up in some ciliated cells and some type 2 pneumocytes, and overlapped in those cells with TMPRSS2. But as you can see here, by looking at all of the points that are negative in blue and the points that are positive in black, really, it was very few cells. And it turned out that this was the case as we looked across many of the different single cell data sets, or atlases, as we like to call them, that we had generated in the past. So in the nasal epithelium, we saw that there were secretory goblet cells and ciliated epithelial cells that looked like they were likely targets in the lung, type 2 pneumocytes and ciliated epithelial cells, and the small intestine absorptive enterocytes. But the frequency of these cells was really low. So, maybe 1 or a couple percent ACE2 positive, except in the gut. And then double positives that had both of the RNAs that we thought were important were even lower. And this was sort of at striking odds with these high levels of infectivity and the levels of virus that we were seeing across the different tissues. And so we began to wonder what was going on. How could you really get this rapid and progressive disease with so much virus when it's really needles in a haystack? And so, this is great, but really told us that maybe we didn't have the full story. And that's because we don't know the actual targets of infection. We only know, what they might be. In parallel, when we were looking at our data, there were a series of beautiful studies that were done looking at, some infections ex vivo, or looking in model systems, and some additional data that started to suggest in models that there were important roles for the interferon response and for certain inflammatory cytokines. But really, the big problem that we came up against was that there were a lot of open questions around sort of, the relevance of these in vitro models and really, what was happening in vivo. And I think that that's really where I want to take you today and talk about some of the biology that we're seeing. And so hopefully a lot of this has been background to this point, but really, I think what Chloe and I have been focusing on is really trying to understand what is happening in disease within an individual. So I'm sure you're all, unfortunately, well too familiar with having your brain tickled with nasopharyngeal swabs for looking at whether or not you have SARS-CoV-2. And usually these are used to do detection by qPCR. What we wondered, because this was sort of a ubiquitous way of sampling, was whether or not we could collect more, given that many of our single cell technologies only require low inputs. So since cells get sloughed off in the process of rubbing back against your nasopharynx, we began to wonder whether these could be equally good sources of cells that we could then use for single cell transcriptomics to identify what are the component cells that show up in viral positive individuals, and also which ones harbor viral RNAs themselves. And so to make a long story short, we spent a lot of time developing a processing protocol that would let us take these swabs, isolate out individual cells from them, and then go process them. And at the end of the day, you get a nice diversity of different cells out of these systems. And so here I'm showing you some immunohistochemistry where we can find goblet cells and ciliated cells, among others. And our full protocol is available here on Protocols.io, and we're happy to help in any way we possibly can. But just think now we have a very low input way of sampling and characterizing the cellular content of the nose, which is going to give us a window into what might be happening at the earliest points in infection, because this is probably the most likely target of where you would begin. So with this profiling pipeline in hand, we went and we set up a cohort of about 58 individuals, some of which are healthy controls, some of which were in the ICU but for non-COVID, but had to be ventilated. So these are severe disease, but not COVID related, as well as some individuals that went on to have mild or moderate COVID. And you can think of this as sort of being WHO classifications 1 to 5, as well as a number of individuals who went on to have severe or critical disease in the sort of 6 to 8 range. And so our controls are going to be, healthy individuals who are SARS-CoV-2 negative, or the ones who were in the ICU who needed ventilation. And then we're going to have these 2 groups of individuals. And this was done as a large collaborative effort with Sarah Glover and Bruce Horowitz and Jose Ordovas and a number of individuals, the University of Mississippi Medical School. And the demographics that these samples represent, in case you're curious, are representative of the communities the hospital serves. So a good range across genders and across ages and across race/ethnic groups. So out of these 58 swabs, we were able to collect about 32,000 cells. And on the right, I just want to show you sort of the map of the different cell types that we collected. This is very similar to the plot I showed you at the beginning, where each of these individual points that I'm pointing out right now is a cell. We've clustered them based upon similarity in their gene expressions, or this high dimensional space of 30,000 genes. And now we are going in and we are annotating them based upon some of the genes they express. And so let me just orient you really quickly as to what we're seeing here. We're getting a number of epithelial cells, which are sort of the lining of your nose right here, ranging from basal-like stem cells to more differentiated cells that perform essential functions like ciliated cells. We have some squamous populations over on this side, which are likely coming from deeper sampling in the nasal pharynx. We have a series of different immune populations that are popping in as well as for some of the aficionados in the group, some populations that have recently been described like ionocytes and enteroendocrine cells. Now that we have sort of this map of what we're seeing here, you might ask, well, are there any major differences that jump out when you look at SARS-CoV-2 positive versus negative? And the answer is yes. If we look here and we overlay the people who were positive for SARS-CoV-2 by PCR with ones that are negative, what we see is that the majority of places where there's this difference is in this epithelial set of cells right here. So a ton of activity going on where we see a bunch of red cells, but not blue cells and vice versa. And, not totally surprising, this is sort of telling you that the viral infection is impacting the cells that we think are likely targets and impacting the barrier. But it gives us a good reason to try and go and focus a little bit more deeply on what might be happening among the epithelial cells themselves. So what we can do is we can remove the non-epithelial cells, the immune cells, and recluster and replot all of our data like I'm showing you right here. So this is really us focusing on the epithelial lineage, sort of that lining of your nose right now. And so we get many of the different populations that we'd expect based upon an understanding of the tissue parenchyma. We've got sort of these basal cells that are stem-like, all these other differentiated populations that I'm talking about in the past. We can begin to ask ourselves how these cells are related to one another, and whether or not they are following normal epithelial differentiation using things like RNA velocity. I'm sure many of you are familiar with this, but the basic idea is that by looking at the ratio of spliced to unspliced RNA, we can figure out what are new transcripts and what are older transcripts, and it gives us an idea of where a cell wants to go. And when we overlay this on top of our data, it follows the general trajectories that we'd expect, where if we overlay the simple cartoon, we have these basal-like stem cells that go up into these more differentiated lineages that include secretory cells and goblet cells over here, move through some of these intermediate cells like deuterosomal cells and over to these more differentiated ciliated cells on this side, with our ionocytes and our squamous cells being separated. So, we're capturing what seems to be the full spectrum of epithelial cells and the interrelationships among them. Great. So you might then ask yourselves, well, what is really changing as we go into SARS-CoV-2 infection process? And so we can begin to ask ourselves as we compare people that are viral positive to viral negative. So the first thing that we see when we look at this is that in those people in which we have SARS-CoV-2, there's an incredible change in the amount of secretory cells that we have. So we see this incredible expanded diversity within here. And so if we look at the percentage of our sample that is secretory, we see a major bump, particularly in our severe and critical individuals, and also in our mild and moderate relative to healthy. As an independent confirmation of this, we developed a flow cytometry panel so that we could take nasal swabs, sort out some of these secretory cells. And we saw once again that in an orthogonal set of samples that there was a significant increase in the frequency of these cells in those people who went on to have the most severe disease. Similarly, we can see that there's a depletion of ciliated cells, so a shift in sort of the overall nature of the ecosystem. And there's also an expansion of some of these precursor cells, where we move from having some of these cells that are important for function, like goblet cells, to more of these durosomal intermediate populations right here. So really what we're seeing is overall an increase in the diversity of the epithelial compartment and a shift where we're losing some of our important functional lineages right here that may secrete essential compounds to protect against infection and to create an intact barrier. And sort of these stress responses where we're trying to generate new things to help us deal with this viral infection. Now, this is sort of at the level of what's happening in terms of the relative frequencies of cells. Another question we can begin to ask ourselves is, does anything change within any of our specific populations themselves? We know that there's a change in abundance, but is there actually a change in intracellular programming that goes with this? And so, for example, let me just show you what we see if we look at our mature ciliated cells in specific. So if we take our controls, so our WHO negative samples, and we compare them to those samples that are either mild, so the WHO 1 to 5, or severe, what we can see is that there's a significant upregulation of a number of genes in response to infection, not surprising. And when we go and look at what is being upregulated, we see a strong enrichment for a number of things associated with interferon signaling, both type 1 and type 2. And this shows up in both mild and severe disease. But intriguingly, if we now go and say, what is different about the people who have more mild versus more severe disease? What we see is that actually there's a much stronger interferon response, both type 1 and type 2, in the context of those people who have mild disease versus severe disease. And we can see upregulation of many of your favorite genes potentially here, like STAT1, a major transcription factor that plays a role in this process, as well as various IFITs and MX1. And I'm showing you this for ciliated cells, but I want to emphasize that this is not just something that shows up in the context of the ciliated cells. It's actually something that we saw across all of our different epithelial populations. So if we look at different goblet cell subsets, or our secretory cells, or our durosomal cells, or our ciliated cells, in each instance, we see a significantly stronger type 1 interferon response in our mild and moderate cohort relative to our severe cohort. In each of these instances. And, this isn't something where I'm just showing you it by cell, which I'm plotting right here. We can also look at this by donor, and we see exactly the same thing where, the people that go on to have mild or moderate disease have a significantly stronger, interferon response. Here I'm showing you by looking at the levels of STAT1 relative to those who go on to have, severe or critical disease. And I want to highlight that this is not something that simply has to do with the timing of when we sample. Actually, if we look at the individuals who go on to have severe disease, we find that, if at swab sampling time they had mild disease and not yet severe disease, they still had this downregulated interferon response, suggesting that really there is in more severe disease this dampened interferon response and interferon responsive gene, or ISG, activity. So you might ask yourselves, well, if you're not seeing a lot of this, who's producing interferon? Are we just not getting it? And so we went back and we tried to figure out who might be producing some of these cytokines that are essential for generating this activity. And when we look across our multiple different cell populations here on one of these dot plots, where these dots show you the percentage of cells expressing, and the color shows you the amount of expression that we have, really, we're seeing very little expression of type, of interferon alphas or interferon betas in here, and limited expression of gamma, with it really only coming in some of our T cell subsets. And, I know that in a lot of places, there's talk about dropout in single cell data because, these are transcripts that might be hard to capture. And so you might just say to yourself, well, is this a technical issue associated with some of the processing that you're doing? We don't think it is. In some parallel studies that we've done in the context of flu with some partners at UMass Medical School, we've seen that we can robustly detect interferon alpha and interferon beta in many different epithelial subsets when you're looking at flu infection. And so we think that this is really something that's more specific to COVID. And this is in line with what a lot of others are seeing, where, in the context of, say, for example, cell lines, where there's exposure to influenza or to SARS-CoV-2, that there's a very mild induction of interferon in SARS-CoV-2 relative to something like flu. And this is not only in cell lines, it's also seen when you look at plasma of individuals. So I think what we're saying is that it looks like, that this virus, does a good job of not inducing this host response, but the degree of response seems to be very important for clinical course. And this is an agreement with what, for example, has been seen in Max Krummel's lab and by others, where, in the more severe disease cases, there is this reduction in interferon in the periphery in different subsets of cells. And so the data that we're seeing falls in very nicely with this. But rather than being in the periphery, it's actually in the primary target tissue where you sort of get this earliest degree of infection. And so, another question might be, what is driving this reduced interferon response? We don't fully know. There's many things in the literature that suggest potential explanations. One, for example, that you may have read about is the idea of autoantibodies against interferons. And so it seems like, 1 in 10 people seem to have, antibodies against interferon that actually can lead to more severe disease. It turns out in our cohort, this doesn't explain our data. We have something that's very similar. 1 out of our 10 individuals, this square that I'm showing you right here, seems to show autoantibodies against interferons, where the majority of our other individuals, seem to show similar levels, regardless of whether they're healthy, mild, moderate, or severe. Another explanation that's been suggested is, some host genetics that may play a critical role. So, some loss of function variants. And these are estimated to be about 3% of the population. So we don't think that it really explains our data, but we're going in, we're doing some additional validations to confirm that that's in fact the case. So a lot of what I've talked about so far is sort of what the virus does to the tissue and the changes that we're seeing. What I want to finish on at least the nasal swab stuff, so I want to talk very briefly about what the virus is actually targeting, because that was sort of the initial question that we had. If you remember at the beginning, I was showing you very quickly that we were using some of these existing cell atlases to try and figure out what were likely targets in the nasal epithelium of SARS-CoV-2. But now that we have this primary data, could we figure out which cells are actually infected and how those cells are changing as a result of infection? And so this is something we can actually do with our nasal swab data for one very simple reason. Just like our eukaryotic mRNA, SARS-CoV-2 has a polyadenylated genome. So what this means is that when we go and try and capture transcripts within cells, we get viral RNA at the same time. And so if we go and we look at our data, what we can now do is we can plot for each of our individual cells what the expression of viruses is, the number of unique molecular identifiers or transcripts that we count per cell. And what you see here is that about 3% of our cells overall are positive, and we're getting good, robust detection of viral positive cells in both our mild and moderate individuals and in those that are severe and critical. Now, before we get too excited and start to analyze this data, we want to make sure that we really believe these calls and that we really believe these are infected cells and that our assay has good sensitivity. So one of the things that we did with help from Pardeep Spetti's team, among others, is went in and did some comparisons between what we were detecting by single-cell RNA sequencing versus qPCR. And what you can see is that we get a very nice linear detection curve with good correlative agreement, where the abundance that we estimate matches up very nicely with what you would get by a standard clinical test. The other thing that we had to spend a lot of time doing, I'm happy to talk about in questions, is cleaning up the data. As you might imagine, one of the biggest issues in figuring out true viral positive cells is distinguishing RNA that's extracellular from intracellular, because you have many virus particles floating around, and you also have cells that are dying and damaged that are spewing out their contents indiscriminately into the environment. And so there was a lot of work that we did to try and clean up viral ambient RNA contamination using some approaches that were developed in some of these papers at the bottom that we played a small role in. And what that did is it led us to a point where we had very high confidence cells where we could say these are viral positive. And as a third layer, to try and get even a little bit further and convince ourselves of what we were really doing, we tried to take advantage of, of understanding of the viral life cycle to figure out which ones were actively, replicating viruses. So as I'm sure you all know, SARS-CoV-2 is a single-stranded positive strand RNA virus. Like other coronaviruses, it replicates by synthesizing a full-length complement of its negative strand, which is then the template for additional positive strand. It also generates a series of nested subgenomic RNA species that encode structural proteins. And further, these things are generated by splicing events where a 5' leader sequence joins onto a 3' body sequence. And so what we can do is we can start to actually look at where our reads in the virus fall. So mapping them to some of these splicing events to positive or negative sense, and so on. And what this lets us do at the end of the day is walk away with an idea of which cells we think are really bona fide infected cells. And that's because we have, high viral UMIs per cell, post-ambient contamination. We've got good coverage of the viral genome. We've looked at negative strand splicing. We've also got our negative strand detection, as well as a series of, spliced RNAs. And so, we find that some of our goblet cell subsets, some of our, KRT13 high helic-like cells, some squamous cells, and developing ciliated cells, and some ISG high ciliated cells seem to be our real targets. You might say, how well does that correspond to what we would have predicted? Well, actually, we do a pretty good job here based upon our original estimates, where we look at the percentage of ACE2 positive cells versus the numbers that have viral RNA. The only ones that are very different are macrophages, and Chloe will return to this, but these seem to play an important role in clearing of infected cells and seem to be, have less processed RNA, and so we think that that might be where they're coming from. The last thing I want to touch on really quickly is like what we're seeing that's different between infected and uninfected cells. And so what we can do is we can look at genes that are higher in those cells that are viral positive versus those that are viral negative. And when we do, we get a series of genes that are upregulated in the cells that are viral positive and some that are upregulated in the bystanders. And many of the biology here makes sense. If you start to look at some of the genes, you can see a number of interferon responsive genes, for example. And so here you see, enrichment for things like antiviral defense, antigen presentation, cholesterol biosynthesis. But interestingly, when we look at what's upregulated in bystander cells, we see strong induction of interferon receptors, suggesting they may be poised to protect themselves. And also, antigen presentation via MHC2, which lines up with some observations that people have made in the context of Ebola too, about the importance of some of these factors in viral protection. You might say, well, if we constrain ourselves to just viral positive cells, are we seeing the same things that have to do with interferon? And the answer is yes. We see surprisingly that there's a similar amount of viral positive cells in both mild and severe disease, regardless of whether we look at positive strand or negative strand. And that really, even in this limited set of cells that have virus, we're seeing much stronger induction of these interferon response genes in mild, moderate versus severe. And on the other hand, what we're seeing in those that are more severe is stronger upregulation of inflammatory responses, which I think will become critical as we think about the next step. So, really brief on this sort of stuff. What we've generated is a big resource of COVID-19 swabs, and it really has given us this idea of the full epithelial trajectory within the nose, an idea of what COVID-19 does really by increasing sort of diversity in the epithelial compartment, but removing some functionality. We've developed some methods that help us identify which cells are infected. And the big thing we've taken away from this is that severe COVID is really associated with this muted interferon signature or interferon response signature that we're really trying to get the origins of, so that we can figure out, what might be predisposing people to more severe disease. But it does seem to be predictive, even at this earliest phase. And so this is what happens at sort of the initial site of infection. The next question that we really wanted to know how does this relate to what happens in other target tissues, particularly to what happens in the lung as you go into more severe disease. And for that, I'm going to step out and I'm going to turn it over to Chloe.
So building on Alex's and colleagues' amazing nasal swab study, Alex and I, and as well as colleagues through MGH, Brigham Women Hospital, Beth Israel, MIT, the Ragon and the Broad, came together to perform a COVID-19 autopsy study, which was recently published in Nature. And so here we're going to share with you some of the highlights of this study. And we're going to discuss some of our adolescent effort, particularly focusing on the lung. I'm going to highlight some results where we compared disease versus healthy tissues to understand transcriptionally and molecularly the main differences observed in COVID-19. And we, like Alex just presented, also look into mapping which cells in particular were infected and how was the infection process impacting gene expression programs. And so across 3 main hospitals in the Boston area, we have performed 13 autopsies, have collected an average between 5 and 14 tissues per autopsies, and have biobank over 400 specimens. And so today we're mostly going to discuss the lung results. And so this is an overview of our COVID autopsy cohort. Every line is a different patient. And I'm going to walk you briefly through the table. Every column here is, one of the organ we might have collected from, and if there's a dot, it tells you we have collected from that particular tissue. And so you have the 17 patients here. Rapidly, you can notice that we have a higher prevalence of African American and Hispanic patients, which perhaps is reflective of the disparity in some of the greater Boston area. We also collected across quite a range of age. Most people may think that patients that were passing away early on, because I should mention most of these autopsies took place between April and June last year. May think that most people were elderly, but in fact, we had quite an age range going from early 30s to over 89. Every patient had a form of long COVID pneumonia, and several had multiple organ failure. You can also see we have a range of postmortem interval, and for safety reason, for 2 hospital sites, we're only allowed to collect samples between 8 a.m. and 5 p.m., hence the fact that we had longer postmortem interval, whereas one of the hospital sites could collect through biopsy procedures, hence the smaller postmortem interval. And so we had a whole process set up where tissues would be collected on the hospital sites and then transferred safely to the Broad Institute, where the tissue would be further processed in the BSL-3 setting, following these different pipelines. So either tissue was processed fresh, dissociated to generate directly single-cell RNA sequencing data, or samples was slow frozen in cryopreservative, which would enable us later on to dissociate and perform single-cell RNA sequencing. Most of our workhorse ended up being flash freezing biopsies and performing single nuclei analysis. This flash frozen sample would also enable us to do bulk RNA sequencing and viral qPCR. Finally, remaining large tissue sections were fixed for different type of microscopy analysis. And so starting with the single RNA sequencing results. So just to give you a sense of how we're getting organized, so everything was coordinated between the hospital and the Broad through a Slack channel. Myself, I was at MGH, and so I would bridge between MGH and the Broad Institute. We would prepare vials that were shipped at the hospital sites and shipped back to the Broad. And here you have 3 of our heroes, Carly, Zohar, and Tony, in their BSL-3 suits that championed the sample processing. And to give you a sense of what the tissue looked like, these are pieces of the lung from the youngest donor to the older donor. And so you can see, if you're not used to looking at this type of tissue, the tissue is pretty necrotic. And that's perhaps expected for a few reasons, of course, because of the long postmortem interval, but also because of the infectious process, you get more necrotic tissue. And so in that sense, that's going to lead us to our first computational challenge, which is dealing with what necrotic tissue can bring in terms of noise in your data sets. So in terms of the computational workflow, after removing ambient RNA, and I'm going to have a small vignette about it in a second, the workflow split into 2. We had the human portion of the RNA analysis to kind of look at the different cell composition in the lung, and a SARS-CoV-2 RNA analysis to try to map the infected cells. And so we're going to start with the human RNA analysis. And so just as a reminder, if you work with very fresh tissue, the odds of you having a lot of dead cells, which contributes to ambient RNA contamination, are smaller. But when you work with tissues from an autopsy setting that took place, in average 18 hours after patient passed away, what happens is that in addition to capturing drops, because most of the data I should mention was generated using the Genomics platform, in addition to capture your cell, you may be also capturing again RNA coming from some of the cells that passed away. And these are examples of pictures from another study that show that you can get debris in your cells. And so how do you get rid of this noise? Fleming here developed a tool called CellBender. And so CellBender is a deep, which is available on GitHub. It's a deep generative model. So what it does is that it learned the background of your RNA profile and can then distinguish cell-containing droplets from empty ones, and then can model to retrieve background free gene expression profiles. And this allows you to boost the magnitude and specificity of differential gene expression across cells. So I'll show you an example here. These are data from the lung before CellBender. You can see all the clusters merging with each other and looking at the gene expression of a particular gene, right? The darker it is, the more a gene is expressed. You can see that gene looks like it's expressed everywhere. But after CellBender, we can now more clearly distinguish our clusters as well as a gene expression profile. So we spent quite a bit of time cleaning up the data with the computational team. The second challenge was related to subtype annotation. And so, grossly every 17 patient samples contributed to every clusters, which is what you're seeing here in the graph. We had 17 broad clusters and every color represents an autopsy sample. So that's a good news. And so we proceeded with two main strategies for annotation. After doing batch correction, we either used automated classifier-based annotation or went into the traditional manual annotation, by which we iterated clustering and subclustering based on specific lineage, identified specific genes that expressed, and used some of the legacy knowledge published to help us annotate our data set. And we wanted to compare the two different approaches. So they were done by two computational postdocs, Graham Hamburg and Raquel Norman. And so in terms of the predicted annotation on the left, Graham used 6 public data sets for a total of 1.8 million cells. These were long data sets across fibrosis, COVID, and healthy. And he classified every single cell using a logistic regression model. And he tested the validity of his training set by withholding 2 of the data sets. And Raquel here on the right used the traditional manual annotation approach. And globally, I can tell you that all lineage matched, but we started seeing more differences when we looked at subclustering or subpopulation of any cell lineage. Here I'm showing you myeloid cell lineage. And so the advantage of the predicted annotation versus manual annotation level is that the resolution in the predicted annotation is at the cellular level, and you don't need any prior knowledge to do this, and it's extremely fast. But the problem is that there's limitation in terms of identifying new populations, right? If some populations that we see in our data set have never been reported anywhere, there's no way the predicted annotation will be able to identify them. So this is some of the hurdles we encounter. So these two methods were quite complementary. And here you see the correlation between the automatic predictions and the manual annotation, and globally, most of course, of the lineage agreed, but as you can notice, some of the subclustering didn't agree. So for all the subclustering, we rely on manual annotation, and identified 7 populations of lymphocytes, 6 populations of myeloid cells, and 5 populations of B cells. The next stage, we wanted to compare disease versus healthy tissue and wanted to know if there were specific cells that varied in abundance and if there were differences in transcriptional signatures. And so here, the analysis was done with our data set compared to healthy data sets. And so what we saw in our COVID samples, we saw an increase here seen on the left, with COVID-19 being in red, an increase in dendritic cells, macrophage, NK cells, fibroblasts, endothelial cells. And we saw a decrease in some of the epithelial cells called AT2 in the lung. They were quite drastic. And so we wanted to look a little bit more into these AT2 cells. And so for those of you who don't think about lung epithelial cells, just as a quick reminder, right, in your alveolus where you have all the gas exchange going on, you have the AT1 cells that are the key cells that enable you to breathe and that perform gas transfers. And the AT2 cells, they're the reservoir that can differentiate in AT1 cells, but importantly, they're also the one that secretes surfactant, which is a substance that is really key to decreasing the tension in your lung to enable the gas exchange. And so we compared the gene expression of the AT2 cells in a healthy lung versus COVID lung, and this is what this volcano plot is reporting. And so what we noticed is that in COVID, in these AT2 cells, there was a decrease in surfactant expression and a reduced level of anti-apoptotic activity. And we saw an increase in sets of genes related to inflammation, innate immune response, damage response, and cell migration. Looking a bit more into the epithelial cell data here, I'm sorry, my clicker is very sensitive. Here you have the epithelial cell subclustering, and we noted that one of the subcluster, cluster 7 here circled in red, was capturing a group of cells called PATS, or pre-alveolar type 1 transitional cells program. And these cells are very key because it's a transition, as I'm going to show you in a slide, it's the transitional states through which AT2 cells differentiate into AT1 cells. And so we could clearly define them based on unique gene expression signatures shown here. And we noted that these particular cells were significantly more abundant also in COVID-19 patients compared to healthy patients. And so these PATs, this program seems to be induced during lung injury, which could be consistent with the fibrosis that is observed in COVID-19. And we saw that it was also associated with an accumulation of another type of stem cell called intrapulmonary basal-like progenitor cells, which are known to act as an emergency cellular reserves for severe damaged alveoli. And that was interesting to us because this had never been reported in human. It's been reported in the context of influenza model in mouse. And so what we're concluding from what our initial observation is that in the context of COVID-19, COVID-19 seems to be impacting the epithelial cell regeneration pathway. So in normal setting, you have AT2 cells here that can self-renew, and they can differentiate, as shown here, through PATs into AT1 cells, which is what is modeled here. Whereas, to summarize, in COVID-19, the AT2 cells, self-renewal and differentiation in AT1 is actually inhibited. And so this transitional states, PATs, ends up accumulating. And we then see these IPBLP progenitors being recruited in the alveoli as a reservoirs for AT2 cells. The third challenge is related to detecting which cells were virally infected and trying to map which gene expression programs were induced by infection. And so we could map most of the reads to the entire SARS-CoV-2 genome, though we had a bias towards the positive sense alignment. And so every column here is a different patient. If there's a red dot, it means we could detect SARS-CoV-2 IMES. And on the left, you have the data before we applied a cell bender and removed the noise. And sorry, this is on the left, and on the right, you have the data post a cell bender. And what you can see in a single glance is that we couldn't detect SARS-CoV-2 IMES in all patients. It was already heterogeneous just from looking at it. And this heterogeneity and inter-donor variation was confirmed by RT-PCR. We also noted that the viral load observed across patients was negatively correlated with the symptoms of onset to death, as shown here. We perform, in collaboration with Participates Lab, bulk RNA-seq of 9 complete viral genomes from these 9 donors, and noted that all donors carried the D614G allele, which was commonly observed at the time in the Boston area. And we also found no other common respiratory viral infections across these patients. We wanted to know, I showed you there were 5 patients that had some SARS-CoV-2 IMES detected, and we wanted to know if they were enriched in this particular cell population. And indeed, we saw enrichment in both macrophage population and endothelial cell population. So every black dot means we could detect some viral IMES, and the more red it is, the more we had an enrichment. And so focusing on these 2 lineage, macrophage and endothelial cells, we knew there were many subsets, so we wanted to know if there was a particular subset within them that were more enriched for these viral reads. And indeed, back to our subclustering in the myeloid cell space, we identified 2 main populations that were more enriched for these viral reads. We should note that very few cells, these are all the different subclusters in the myeloid cells, had SARS-CoV-2 detectable IMES. However, this was significant. And comparing virally infected cells to non-virally infected cells, we could find several interferon-regulated genes upregulated. Similarly, for endothelial cells, we also looked across all of our subclusters and identified two main subpopulations of endothelial cells that were enriched for these viral reads. Similarly, we could detect only few cells within these subpopulations, yet this was significant. Taking a few step back, like similarly, we find similar differentially expressed genes. And taking a few step back and looking at all the myeloid cell populations, if you look at those that were virally infected, as highlighted here with these SARS-CoV-2 genes, we could detect enriched in the RNA positive SARS-CoV-2 cells, several interferon-regulated genes, as well as key chemokines involved in attracting lymphocytes. We also wanted to know where were the viral entry factors like ACE2 were expressed. And so here you have a list of them color-coded, and you can see that they were mostly expressed in epithelial cells. Yet, as a quick reminder, this is not where we saw our virally enriched SARS-CoV-2 reads. And so we use orthogonal methods to look into this, such as macroscopy. And here, imaging of the infected tissue compared to non-infected tissue, where you have in DEPI, you send the nuclei and you have SARS-CoV-2 in pink, we could detect SARS-CoV-2 in epithelial cells. And we reasoned that the reason we couldn't see it in our single nuclei data is that when we dissected it, one of the reasons could be that when we dissected the tissue for downstream single cell analysis, we excluded some of the necrotic tissue. Remember, I showed you the different pictures. And the necrotic tissue could also be enriched for some of these epithelial cells that have been infected. So that could be one of the reasons. To better understand the tissue pathogenesis, we also use special methods, which are complementary to single cell RNA sequencing approach. And here we use the nanostring technologies to do this, by which we can stain the tissue, look for pentaK positives to look at more epithelial cells, to pentaK negative tissue, select specific region of interest, here displayed by these circles, and then sequenced the transcriptional profile of these regions. We profiled 14 donors, including 3 healthy tissues. The region of interest spanned different anatomical structure, viral abundance based on our initial SARS-CoV-2 RNA hybridization signal. And then we segmented the data based on pentaK positive and negative signal. And so the convolution of the major cell type composition across these different tissues, we noted that for pentaK positive compartments, so the epithelial cell compartment was mostly dominated by 81 and 82 cells, whereas we saw greater cellular diversity across the pentaK negative cells. Comparing the COVID-19 alveolar region of interest versus the control, we saw several pathways that were upregulated in COVID-19, including interferon alpha and gamma response and oxidative phosphorylation, just to name a few. And this is for the pentaK negative, and for the pentaK positive, there were similarly the same interferon gamma alpha response upregulated. We saw also IL-6 and JAK3 signaling upregulated, which is interesting if you think about this tocilizumab trial. Finally, as some of you may know, there's an international effort looking at genetic risk variants, making more susceptibility for COVID-19. So we sought to use our ATLAS data to try to understand the cellular context of the genetic variant. This is a nice slide provided by Alex to remind us that actually genetic variants can influence the different transcriptional profile for our cells. And so out of 26 genes that are proximal to 6 of these GWAS COVID regions, which are displayed here, we had 21 genes that were specifically expressed. So if it's red, it means that it's highly expressed in one of the specific cells. And so, for example, FOXP4 was specifically expressed in 81 and 82 cells, or CCR1 and CCRL2 were specifically expressed in macrophage. So that helped us start mapping the cellular context of this genetic variant. We also look if some of these genes were differentially expressed in COVID-19 versus healthy lung, these same genes, and identified 18 out of 26 that were. So for example, this transporter here is significantly overexpressed in goblet cells, or CCR5 significantly overexpressed in CD8 T cells and Tregs. This is just a summary plot here, but the highest number of driving genes that we observed were all in lung 82 cells, and these span several loci, which could potentially point towards a more polygenic architecture linking 82 cells versus with severe COVID-19. And most of the implicated GWAS loci, they were close to genes included, for example, OAS3, which is a key IHGs in lung AT2, and as well as in club cells. And there were several other unresolved GWAS loci when we don't know the exact involvement, but included these three genes here. So obviously our data set is very small. There's a limit to which we can do data interpretation, but it can help set some foundation to understand cellular context. Some of our ongoing analysis include analysis of the liver, kidney, and the heart. For what it's worth, so far we haven't detected any viral transcript in these two different organs, and this was confirmed by RNA-scope and nanostring. And so in summary, we've highlighted you some findings for our lung atlas, showed some specific transcriptional differences between healthy and COVID-19. I've highlighted how COVID-19 really has a strong impact on lung regeneration. We have identified specific cell types and subsets in myeloid epithelial cells and endothelial cells that are enriched for viral reads. And I've shown the cellular context of some of these GWAS hits. And if you want more information, I invite you to look at the manuscript. And Alex, I'll let Alex jump in here.
Thanks, Chloe. I'll just finish really quickly by saying, a lot of what Chloe's lab and my lab and a number of others have been trying to do is to really understand, the cellular basis of disease within humans. And a critical part of, of once we've identified these things is then figuring out what are the appropriate model systems to use to do this. And so, a counterpoint to this has been characterizing multiple different tissues, cell lines, organoids, and surrogates that we could then use as a way to actually go and test interventions to accelerate back into humans. And so in concert with everything that we've been doing with the human samples that we've shown you, we've been running matched samples in the context of non-human primates to look, for example, at the efficacy of vaccines, or in Syrian golden hamsters, which represent a good model for severe disease. And I'll tell you that many of the responses that we're seeing in these models faithfully recapitulate aspects of disease that we're seeing in humans. For example, major changes in the epithelial compartments in the lung of the Syrian golden hamsters and in some of the immune populations that come up as critical in the monkeys. And so I think we'd all just like to take this time to conclude. These are studies that are not something that that anyone lab could do. They're large collaborative networks. We first want to thank all the study participants and their families, without whom none of this would have been possible. And we very much appreciate their willingness to partner with us in these studies. These are huge collaborative efforts, particularly challenging, as Chloe pointed out, in the context of the pandemic. And so it was incredibly amazing to see all these hospitals and scientists come together to drive this stuff forward. And obviously, thanks to all of our funders who've helped support some of this work. And we're happy to take any questions. Yeah. Sorry, go ahead, Chloe.
I know, I apologize. It looks like our slide about data portal has disappeared, but Alex and I had a slide about data portal, so we'll make sure to pull it out. So all our data is publicly available, as we want to help the community to leverage our data sets for their own respective research. And I'm happy to take questions.
Alex, Chloe, that's impressive. That represents a lot of work. I've got a lot of questions. Audience, either type your question into the Q&A box and or raise your hand. Let's see, we'll start in the nose. Alex, why do you think there's ends up being this huge population of secretory cells in the infected population? Is it that the ciliated cells are dying in them, or is it changing the developmental program?
So I think that it's a stress response. I think it's a great question. I think that in a lot of places we've seen that epithelial populations can differentiate and de-differentiate in order to try and deal with stresses. And this happens in the skin, in the nose, in the lung, in the gut. For example, as Chloe was showing with sort of the attempts to replenish some of the epithelial populations in severe disease. So I think what's happening is you have, an environment that's full of different PAMPs and DAMPs, and it's causing some of the surrounding epithelial cells to try and do what they can to restore barrier integrity and to provide some modicum of support. And I think that one of the issues is that it compromises overall barrier function in the process. So trying to figure out ways to secrete things that will help protect, to get rid of stuff. So not surprising, you see a lot of diversity there. And then, some of these more terminally differentiated cells, not surprisingly, you see a dedifferentiation to try and create some of the populations that would be essential to help provide host response.
That makes a lot of sense. I had a related question, and yeah, I always have this question with single cell RNA-seq data. When a lot of the critical molecules are secreted, it's, trying to capture the role of the ligand and receptor. So like one specific question I had is, and we've seen this from other research as well, that it, on the ligand side, it seems to be mostly interferon gamma, but then the receptor that you saw differentially expressed was the alpha beta receptor. So, but what's going on with that mismatch there?
So to be clear, at least in the nose, it looks like in bystander cells that there's an upregulation of some of these interferon, related genes in some instances, like interferon alpha receptor. So not surprising that, given some general danger signals in the environment, the cells would try and, respond by regulating something that can help make them responsive. I think the big thing that we're seeing in the context of COVID that many others have seen is this virus is very good at not inducing interferon responses. And that seems to be one of the reasons why it's so problematic. And in individuals who can mount effective interferon responses somehow manage to do that. They seem to have better clinical courses. Whereas in individuals where that doesn't come up, they seem to be progressing to more severe disease. And so, I think it's not surprising that we don't see a lot of interferon given what everybody's saying and what others have seen in vitro and in vivo and in the periphery. I think what we really don't understand yet, as I was pointing out, is like, why is it that some people produce a little bit or just enough that you get these ISGs and why do others not? And we don't have an appropriate mechanism, but I think that figuring that out is going to be important for thinking about ways to create, prophylactics and therapeutics moving forward. And Chloe, you want to comment too?
No, I think it's perfect answer, and especially important as we start tackling long COVID. How does it have an impact on you as you develop these symptoms long term? But yeah,
we had Ben Tenuver on, and his lab is trying to figure out which viral ORFs are shutting down the early interferon response. I dimly remember it was ORF8 through some unknown mechanism, but I may be wrong. Sarada, you want to jump in?
Yeah. Thank you, Alex and Chloe. A question, Alex, is probably for you. Could you please speak a little about the cleaning the data? I mean, how do you differentiate between the actual viral RNA and RNA from the infected cells?
Yeah, so I'll add a little bit, and Chloe can as well, since she showed a lot of it in the context of the lung, where, particularly with the long postmortem intervals, it's incredibly important. At least in the data that we were doing for the viral reads themselves, we first want to make sure the assay is quantitative, which I showed you with the qPCR, which is great. We then go and do some ambient correction where we're basically looking at what are the transcripts that are showing up in the environment on things that we don't consider to be cells. So in a lot of these technologies, we have beads that are used to pair to single cells, and not every single bead is matched with a single cell, but they're matched with ambient. So we get many places where we get 4 or 5 or maybe 10 transcripts on a particular barcoded bead, which we usually think of as sort of the ambient background. And so what we can do is we can think of that as a soup, as some things do in like SoupX, and then statistically sample from that to figure out when we believe that a transcript with some probability is likely to have come from a cell as opposed to a background. And so we basically model that, and we overlay a process on it. It was some stuff that we worked out originally, a really talented pair of students in Pardis Sabeti's lab, Erin Lynn and Dylan Cutler, and it's in a paper that we did on Ebola where, there's also a lot of damage and death. And so we adapted that here, and it seems to work well. And then to give us even more confidence, because you don't always know whether, it's actually active infection, we also look at viral life cycle. And because we know a lot about sort of the splicing and, sort of going from positive to negative strand, and then getting into all of the different splice variants, we can look at the alignments to each of those segments to give us a better sense of, whether or not this is a cell that's actively undergoing infection and viral replication, or if it might be a cell that was just, phagocytosed or, virus that was free that was eaten up, because it was bound by antibodies or the like.
And that's particularly important, that latter point, given the observation with the myeloid cells, because there's so much enrichment, but it's not clear to this point if it's just phagocytosis. And the data with so we, obviously the talented student in Alex's lab, Kylie Ziegler, who performed the work for Naser, also performed the work for the lung autopsy, so it's the same process. But the lung autopsy data is very noisy as well, and lots of dropouts. So we only had very few alignment with a negative strand, so it's still hard to know if it's productive infection or not in our autopsy data.
I had a related question. So I thought it was really fascinating that you showed that it was only weak correlation between the ACE2, TMPRSS2 expressing cells and the cells with the highest viral load.
By sequencing, right?
Yeah, okay, and it made a lot of sense that the highly infected cells are necrosing and dropping out. But do you think? I mean, I know ACE2 is really low abundance. Do you think it's just dropping out of your sequencing, or is the virus getting into those cells by other means, or have I misunderstood what you showed?
No, you didn't misunderstand that we couldn't map it based on our single nucleotide and single cell data as of why. Definitely, probably, and Alex can add to it, a mix of, of course, the transcript being more locally expressed, but also perhaps the hypothesis you just mentioned, the cells being infected are more likely to die. And when we got the tissue, it was so necrotic that, of course, we biased ourselves against the necrotic tissue. We went and took the live tissue. And so we probably biased ourselves in the sampling against the infected epithelial cells. And that could explain why we couldn't detect it by sequencing. However, by RNA-scope and also nanostring technologies, we could very well detect it in the epithelial cells. So it is definitely a technology also bias. But also, like, the fixed tissue was handled very differently than the one we used for the single cell and nuclei sequencing.
Yeah,
that's great. The only other thing I'd add is remember that in some places RNA is not necessarily a great surrogate for proteins. You also have to think about how stable that protein is, and how abundant it is, and there are other hypotheses that I have for how some of it may arise, but multiple layers to fold onto that.
Yeah. That's why we try to not rely on a single technology to drive our conclusions also.
Yeah. That's awesome. Sarada, do you have another one? I've got
a quick one. Speaking of necrotic tissue, do you see a correlation between the extent of necrosis and disease progression, and also a correlation between the extent of necrosis and the age and sex of the patient?
That's a good question. I'll let also Alex add to it. I think we need to be careful with our conclusions because the sample size is very small. And also there's a confounder because of the post-model interval, which we couldn't control for. And so with these two factors, it turns out that the patient that were the youngest did turn out to have the best looking tissue. And perhaps that is not surprising if you compare the tissue of someone in their 30s to someone in their 80s, you would expect just from the how status of the tissue to look a little better. In terms of the correlation between the extent of the disease and necrosis, I don't think we can drive any conclusions because of the confounders of the postmortem interval, which had nothing to do with the severity of the disease, more when the patient passed away.
True.
That's a good question. There's a lot of larger autopsy effort that are about to unfold in the year to come as part of long COVID effort. And with we're talking about hundreds of autopsies, and with this type of sample size, we'll be able to better tackle these questions.
Yeah, that'll be interesting to know.
We don't see a strong association, at least in the nasal swabs.
Okay. Thank you.
I thought the GWAS data was super fascinating. I was really struck by the two huge peaks, the one on chromosome 3 that seems to map to FICO1, and then a big one on chromosome 9 that you didn't have that annotated. Do people know what that chromosome 9 peak is?
I didn't annotate it because there's no I mean, Alex can add to it if I'm wrong, but there's no clear genes, right, in that large span that I could see associated with it. And Alex encouraged me, to be honest, to include the Chinois data. I tend to be a bit reluctant to oversell it because our sample size is so tiny, but Alex made a good point that it can at least teach us about the cellular context of the disease. And so, yeah, we don't know the exact gene. But the good news is that there's large effort at the international effort at level mapping these genes. So,
yeah. The only thing I'll say to what Chloe's already added is in a lot of places, we don't necessarily have power on our end, but we're trying to begin to have a window to understand how some of these things may play out to generate hypotheses. And so, I see understanding sort of the cellular sites of some of these particular SNPs as being essential to thinking about how they might function, but it's definitely not definitive. And so we put it in there because I think it helps to remind people that when you think about genetic mutations, you have to think about the machines that operate on that information, so the cells. And so it's a lens to think about these things, but we still have much to do in order to figure out the ways in which those may actually be causative, and if so.
Yeah. And so to the extent of using it, the only way I'm comfortable, like I speak for myself, is just trying to understand the cellular context where these variants may be playing a role. But so to answer that specific question about that peak at chromosome 9, is because we mostly highlighted that when there were genes that were known close to the region, and there's no clear genes in that region.
Yeah. Yeah, and Jean-Laurent Casanova's data seems to suggest that it might be an agglomeration of rare mutations, at least in like the interferon 1 mutations that he found. So
it could be a mixed.
Get clear peaks. Chloe, Alex, this was really amazing. So excited, and thank you for joining us.
Thank you for having us.
Thanks for having us.
Thank you.
Okay.
Take care, everybody.
Bye.
Yeah, take care. Everyone, we didn't have time to make a closing slide, but we'll be back next week, same time, same place. So everyone stay safe.
Bye.
Bye-bye.
You as well. Take care.